Hello everyone, and thank you for joining us for the Medical Research Scotland Academy. This is the third week of 4 virtual events where we will hear from experts across the medical science industry and academia. You will get the chance to meet medical researchers and scientists exploring what their work involves. Today we will hear from Dr Donald Morrison, lecturer in microbiology at the Edinburgh Napier University, on why antimicrobial matters. We'll then hear from Dr Fiona Stainsby, Senior Lecturer in Microbiology, also from Edinburgh Napier University, and she will be discussing microbes, myths, and the environment. So let's get things started. Over to yourself, Dr. Morrison.
First of all, introduction. I work at Edinburgh Napier University, and I've been working with superbugs for over 30 years now. And I'm going to try and tell you my story with superbugs, how I got involved, and why they're important, why they are of critical importance to everybody. It's not just one group of them. But first of all, I'm going to give you a question to see where you are in terms of your knowledge of superbugs. But first of all, tell me how you're feeling today while we wait for everybody to get on board. When I do this with my students at university, and I do it just again to get everybody onto Menti, I get a whole range of dots put on different figures. And of course, you know, we're all different. Some of us are morning people, some of us are evening people, and some of us are all-day people. And some of us have days that aren't quite so good at any point of it. So, okay, that's really good. So the question I want to ask you before I start my presentation is, have you seen much about AMR or superbugs at all? You know, it's been around. I've got a news article there from 2013, so it's been around for quite a while, but you know, it may not be reaching every part. So it'll just be interesting to see where you've seen it. Have you seen it at all? So a number of you haven't seen it at all, and that's really interesting for me because I've been working this. It's fore and center for me in my work, and of course, work dominates life as well quite often. So it's been there focused for me. So it's really interesting to see that there are a number of you that haven't heard anything about it. So for those anyway, it'll be an interesting journey for you. So now, superbugs and why they matter and how I got involved with them. So my life with superbugs, and this is the most terrifying image you can see. It's just an ordinary bacteria doing what it does with lots of flagella to move around, but it looks quite terrifying. And this is me standing proud with my bit of equipment that I used for tracking superbugs in hospitals and elsewhere. It's called a pulsed field gel electrophoresis equipment. It's my kind of pet bit of equipment. So how did it start for me? First of all, it wasn't a planned journey, and I'm sure for most of you, your career will not necessarily be very planned. I first of all went into agriculture. I was going to do a higher national diploma in agriculture, and I got ill, and then I turned to my second favourite topic, which was biology. And I went to Edinburgh Napier University, where I am actually teaching now after 20 years later, to do a diploma. I didn't have the right qualifications or enough qualifications to get into a degree course. So I was probably a slow starter. When I got to university, that's when the light went on and I realized, you know, studying and education is just the best thing there. So I did my diploma. When I finished that, after 3 years, I went to Heriot-Watt into third year and did a BSc in microbiology. This was within biology. This was the area that really got my attention. I then did 2 years in Heriot-Watt, and I went then to do an MSc, and this time within microbiology, the bit that really interested me was medical microbiology. So I went to the London School of Hygiene and Tropical Medicine and did my MSc there. Then I started working, and during my work, so this was a part-time PhD, so it took me 8 years to do my PhD. Normally it takes about 4 years, 3 or 4 years, but I was doing it part-time while I was working at Napier. My first employment was with what was called then the Health Protection Agency. It's now Health Security Agency, and the Laboratory of Hospital Infection, this grand building in London in Colindale. This was my colleague, Neil Woodford, who I spent and still friends with him and still working with him. And here I was working with superbugs in hospitals. I then moved up to Scotland 10 years later and worked at the MRSA Reference Laboratory, which was based in Glasgow at the Royal Infirmary in the Lister building, named after Joseph Lister. And here, superbugs, I was working with them not only in hospitals, but also they were moving into the community and, interestingly, into livestock, into animals. And then 10 years there. So I've done 10 years in each of these employments. And then I moved into academia at Napier 10 years ago. And here my interest in AMR moved to superbugs in the environment. So hospitals, community, livestock, and the environment are the areas that I've looked at AMR. And this is us doing some sampling out near on the East Coast. Okay, so superbugs, what are they? AMR is the other name you find for them. So basically here you've got a group of yellow cocci bacteria, and the majority of them would be killed by the antibiotic. But there's a couple of them which are resistant. And what happens is we treat people, rightly so, with antibiotics to help them to treat infections. And these bacteria then become more resistant as you treat them. So by the end, you've got the majority of them now are resistant. If you treat somebody who's got infected with these red ones, the antibiotic will fail. And if it's a serious infection, the person will die. They looked in caves in New Mexico. This cave had not been inhabited, had not seen a human soul for more than 4 million years. They found bacteria there, as you find them everywhere, and they were resistant to many of the antibiotics we use today. So this was telling us that resistance is natural, it's in the environment, it's there. But what's happened is that we use lots of antibiotics and we select for these resistances. What about deaths? This is deaths attributed to different diseases. Tetanus, you've got here, 60,000 per year worldwide. Road traffic accidents, not a disease, but how many deaths there are due to road traffic accidents? 1.2 million. Measles, diarrheal diseases, diabetes, cholera, and the big one we have today is cancer. So what they've discovered, this was in 2016, with AMR at the moment, well, 2016, 0.7 million people die with AMR-related infections. But if we do not do anything about this problem, that we have this crisis of AMR, then by 2050, there will be 10 million deaths per year. And that's kind of the crisis. That's a scary place we're in terms of AMR. This man, Jim O'Neill, was the one who brought out this report and highlighted it to the UK, but also internationally. So now there's lots and lots of effort going in to stop this scenario of having 10 million deaths per year from AMR. AMR involves all different sectors. It involves the human sector, it involves animal health, and it also involves the environment. But when you've got a crisis, everybody wants to find out who's responsible, who's to blame. And there was this article that I read about, was it human medicine or was it agriculture, the animal medicine? vets that were responsible for this crisis, because the more you give antibiotics, the more resistance you get. So who's responsible? And part of my work, or not my work in total, but I'm involved in this, trying to find out what is causing this crisis. How can we then avert it? The first question was, is it the animals? Is it how we treat animals with antibiotics? Is that what's causing the rise in antibiotic resistance? Should we start treating animals as much as we do? I was working at, as I told you, at the Laboratory of Hospital Infection with Neil Woodford, and this was my first publication, and this is of course important if you're a researcher to get papers published. It was in 1989, and it was on this superbug, vancomycin-resistant enterococci. We had just published this. The first one was found in 1986. And then a few years later, Janice Bates she was working in Oxford, said that probably the reason for these superbugs, these vancomycin-resistant enterococci, their origin is from livestock, from animals. Now, this was a revelation to us. We thought it was just within the hospital environment this was all happening. Lo and behold, they were treating animals with an antibiotic called avoparcin, and treating them because if you give a little bit of antibiotic, the animals will grow to a much greater extent, so therefore better production rates, better levels of proteins. But this is us treating animals with antibiotics. When I was in Glasgow working in the MRSA reference lab, we found similar things. Yes, there was superbugs in hospitals. MRSA was the one I was working with, hospital MRSA. We then began to find them in pets. So where was this coming from was the question, and we realized it was coming from the hospital, patients coming out and giving it to their pets. But then we began to find these MRSA, these superbugs in the community, and we just didn't know where they had come from. We knew the pet ones had come from the hospital, and then we saw that these MRSA were in animals, and we called them livestock MRSA. So this is a whole scenario of areas, and of course, the big health issue is if these MRSAs come from all these areas into the hospital where you've got very sick people, then you will get people dying from these superbugs. Hold on. So the question was, who is responsible for it? Was it the animal sector? Well, there was some evidence for this, but then there was other evidence which said that it wasn't the animals, because other methods that were used said, no, it's not the animals. It's not coming from the animals at all. So the question then was, is it coming from the environment? And this is where my own and Fiona's research has come in, asking, what about the environment? Where is it involved in MRSA? And is this where the problem is arising? So this is a scenario now from wastewater treatment plants, farms, from water, rivers or bathing water, or from wild animals. Is our superbugs coming from these places into our hospitals? And that's our research. We're asking the question, what is the environmental dimension of AMR? And if you ask where they're coming into the environment, well, you come through livestock, from aquaculture, human manure from animals, manure from humans, biosolids, septic tanks, wastewater treatment plants, and CSOs, combined sewer overflows. And this is a picture of the whole scenario of transmission from the environment, from animals up at the top left of the picture, into crops, into the water. From the water environment, you get animals drinking, wildlife, people swimming, and then, of course, coming back into the human population. My final slide is just to show you that there is good evidence that many of these superbugs are originating in the environment. This is a list of 5 antibiotics that are used to treat patients with serious infections. And there's been several studies to ask, where did these resistance genes come from? And for each of these, the answer is they have come from bacteria in the environment. Staphylococcus fluoreti for methicillin, the MECA gene, Pienobacillus and Rhodococcus for the Van A gene, Kluvera for cefotaxime, and this is the one that myself and Fiona are working with a lot at the moment, Schwanella for ciprofloxacin, and for this last resort antibiotic carbapenem from Erythrobacteria littoralis. So here is the evidence: the environment is part of this AMR crisis. And of course, for us, it makes our research not just interesting in and of itself, but it's supporting a very important health issue. Thank you for your attention. I will now be handing over to my colleague Fiona to take you through the rest of this session. Thank you for listening, and I'll hand over to Fiona.
Thank you. Thank you for introducing me briefly, Donald, and thanks to Medical Research Scotland for inviting both of us to take part in this Academy session. I'm delighted to be here alongside Donald. As he's mentioned already, we both work at Edinburgh Napier University, and my role involves teaching and research. I look after PhD students, and I guess I am a microbiologist. I don't have any medical background, though. I would probably consider myself an environmental microbiologist, and Donald comes more from the clinical side. But as you've probably learned already from Donald's presentation, there's a really important link between the environment and human health. And that's just not for antimicrobial resistance alone, but for all sorts of diseases, from cancer to heart disease. And pollutants are probably one of the key things that we need to look at when we're thinking about the impacts on human health. So I'd like to invite you to join this quiz. So it's the same Mentimeter code. So if you're logged in already, that's great. If not, if you'd like to rejoin, if you can, either using the QR code or using the session code, which is at the top of the screen. And I believe it's also in the chat function. And whilst you're joining, I'll just briefly explain what the quiz is all about. So we're going to see how much you've listened to what Donald said already. And we're going to talk a little bit about the use and misuse of antibiotics and how antimicrobial resistance can develop and the risk that that poses to human health. And we'll learn a bit more about how antimicrobial resistance can spread through the environment, and indeed how us as scientists can monitor and look at the things that encourage the formation of resistance in the environment. So let's see what you already know, and we'll try to dispel a few myths about AMR along the way. This quiz is anonymous, of course, so don't be shy, just give it a go. It doesn't matter if you get the answer wrong. If you're not sure, just take a guess. So the first question is quite straightforward. Have you ever taken antibiotics for an illness? You might have to go back in time, and maybe you won't remember because maybe you were given antibiotics when you were quite young, maybe prescribed by your GP or perhaps even a dentist. Okay. So yes, multiple times, gosh, already, and you're so young. So that's interesting. That's interesting to learn. But some of you know never. That's quite nice. That's a good place to be. Okay, so how many times do you think the average person in Scotland is likely to be treated with antibiotics in their lifetime? So this is a sliding scale, 0 through to 50. And I can see your answers popping up. So we've got quite a range here. And that's moving around. So some people think maybe close to maybe around about 5 or 10. Quite a cluster up between probably the 5 and 10%, sorry, 10 times range. not so many up towards 30, and nobody thinks it's as much as 50. Okay, so let's have a look. So 15 is the average number of times someone living in Scotland will be prescribed antibiotics during their lifetime. But of course, it varies with individuals. Yeah, so some people have medical conditions, and other people have very healthy lives and are fortunate enough to not need antibiotics very often. Children typically have antibiotics for common infections like ear infections, chest infections, throat infections. So that's probably why you've been prescribed antibiotics before. As we become adults, other things come into play like urinary tract infections, molar teeth extractions, those kind of things. And of course, as we get older, there's the greater chance that we might have surgery. And older people often suffer from chronic conditions and need repeated antibiotic treatments. So let's just ask ourselves that really fundamental question: what do antibiotics do? So do they kill viruses, boost the immune system, treat infections, or heal wounds faster? So you pick your choice. There's only one option. Oh, well done. That's great. Okay. So there's a difference between antimicrobials, which is a broad term for drugs that we can use to treat all sorts of microorganisms, from viruses through to fungi. But antibiotics, which are also called antibacterials, that Donald's been talking about this morning, only treat bacterial infections. Yeah. And they play a critical role in public health, clearly. Modern medicine would not do very well, particularly surgery, if we didn't have antibiotics to ensure that infections didn't develop in wound sites. But antibiotics only work against bacteria, not against viruses like the common cold. So I hope you've never been prescribed antibiotics for the flu or the common cold, because they don't work against viruses. If you've had antibiotics, you've probably taken them as a tablet or as a capsule, but they can come in liquid form. If an infection is really serious, then it may be intramuscular, so injected into the muscles, or even intravenous directly into the bloodstream, rather than going through the stomach and the digestive system that way, just to get it to the site of infection more quickly. And some people who have lung or pulmonary infections may take their antibiotics through a nebulizer or an inhaler directly into the lungs. So there's lots of ways that we can take antibiotics, and lots of different types of antibiotics. So this question now is maybe hopefully not a trick question, but it's a true or false. So antimicrobial resistance occurs when people become resistant to antibiotics. What do you think? So do people become resistant to antibiotics? Okay, so most people think this is true. Okay, I shall reveal the answer. It's false. It's false. So there's our first myth busted for today. So no, people don't become resistant to antibiotics. We sometimes talk about patients being resistant to antibiotics who are in hospital, but no, it's the microorganism itself that develops resistance to antimicrobial drugs, not the human. So there's no change in us, essentially. It's in the pathogen that is infecting us. That's where the change occurs. So whether it's an antibiotic or an antiviral or an antifungal agent, depending on what kind of microorganism that we are tackling, resistance is absolutely within the microorganism itself, and it can change over time. It can mutate to a point where they no longer respond to the medicines that previously would be used to treat and cure those infections. So if we think now about antibacterial resistance specifically, do you think this is encoded by bacterial genes? Lots of answers coming in, and you're all unified in your agreement that this is true. And you're absolutely right, yes. So humans don't become resistant, the organism does. And in the case of antibacterials or antibiotics, then the resistance forms within the DNA, the genetic code of the organisms themselves. And they have resistance genes that occur there naturally, or indeed can be shared amongst microorganisms. And this is something that we worry about quite often. We think about the transfer of resistance genes between different populations of bacteria, and that can occur in different environments. So it can occur in biofilms in hospitals, it can occur in soil and in water, out in the natural environment, so lots of different places. And it can occur between pathogens and normal microorganisms that normally wouldn't cause us any issues, but then those genes can be transferred and spread to lots and lots of different bacterial populations in the environment. And ultimately, the more that those resistance genes spread, the greater the chance of that re-entering the human population again, and then it being more of a challenge to treat common infections with our normal antibiotics of choice. And there's lots of factors that can accelerate the spread of resistance in the environment. And this is what Donald and I are really interested in at the moment, is what is it in the environment that's causing the spread of antimicrobial resistance or antibacterial resistance specifically. And I've put this slide up just to show you that there are lots of different things that can drive the development of antimicrobial resistance genes and the spread of resistance genes amongst bacteria. And by no means is this everything, okay? So this is just a handful of things here. If we think of the hospital as an environment, there's lots of biofilms that can develop on patients, on their skin, in things like catheters, on surgical instruments, on beds, on curtains, and various other areas. And in those biofilm environments where bacteria are in close proximity to one another, they can share genetic material. They will literally transfer their genes from one cell into another, making what was a harmless bacterium resistant to antibiotics and dangerous. We know that the misuse of antibiotics, so unnecessary prescriptions for viral infections, people not completing the course of antibiotics. So I'm thinking back to Donald's earlier slide, where if you use antibiotics and you don't finish the course, the bacteria that are resistant, naturally resistant to that antibiotic, then get the chance to grow and flourish. And we know that there's misuse and overuse of antibiotics in agriculture. And ultimately what's happening here is that whenever we give an animal or a human an antibiotic, not all of that is absorbed by the body, and any unused antibiotic is excreted back in our urine and our feces, flushed down the toilet, and it ends up back in the environment in one form or another. And then what we see in the environment is higher levels of antibiotics. And this acts as a driver for those microorganisms exposed to those higher levels of antibiotics in the environment to develop resistance. It's an advantage for them. There are other stress factors that can also encourage the development of resistance in bacteria. So things like disinfectants or other biocides that bacteria are exposed to, that are not at doses high enough to be lethal to those bacteria, so they survive. And as a stress adaptation, they can develop resistance through increased mutation rates in their genes. So I guess the message here is there are lots of things going on, and the environment plays a really important role. So another question for you now, where do you think antibiotics that end up in the environment originate from? So this is a word cloud, so you've got the chance to type in between 1 and 3 suggestions of where you think these antibiotics are coming from, and you might have heard me mention a couple of sources already. So you can just type those in and submit them, and hopefully it will come up as a word cloud. Hospitals, absolutely. Where else do you think they're coming from? Yeah, germs, people, water, manure, that's a great one. Wild animals, indeed. Super. Lots of ideas now. Yeah, hospitals is quite popular, isn't it? Still water, livestock, and sewage. Oh, CSO, brilliant. Yeah, combined sewage overflows. Oh, I'm really delighted to see that. You're absolutely right. There are many, many sources of antibiotics that end up in the environment. As I've already said, it comes through human waste. We don't absorb all of the drugs we take. So from our normal domestic toilets, but also from hospitals, but also from pharmaceutical manufacturing sites. Yeah, so that waste will end up in the water too. We're giving livestock antibiotics. Wildlife will also be exposed to antimicrobial resistance. There's things like aquaculture. So fish farms, salmon farms, and so on will use antibiotics to treat fish for all sorts of pathogens. And when all of these antibiotics are released into the environment, they affect the bacteria that are there in the water and encourage them to mutate and develop resistance, which can then spread to other bacteria. So great, fantastic. All of these suggestions are right. And of course, wastewater treatment plants are one particular source that Donald and I are concerned about because we concentrate waste there. And when we do that, bacteria can share lots of genetic material. And one other source is that the solids that come from sewage waste are treated, and often they're used as fertilizers that end up back on the land and used to fertilize crops, thereby sometimes reintroducing antibacterial genes or bacteria back into soil. So there's this whole cyclical effect. So where do you think we have found antibiotic resistance bacteria in Scotland? So there's lots of options here. So fresh waters, rivers, lochs and coastal waters, wastewater treatment plants, crop soils, farm animals and bird droppings. And you can pick more than one answer, of course. Yes, you're absolutely right. They're all right. Unfortunately, we do find antibiotic-resistant bacteria in all of these areas, and to different levels. Some are not concerning, but we do know that treatment plants are another hotspot, like hospitals, where we find a lot of antibiotic-resistant bacteria. But interestingly, birds, so you might have picked that up from Donald's presentation right at the start. So birds can ingest contaminated food or water, and then they can spread those antibiotic-resistant bacteria again through their droppings. And because they fly, and insects are the same, they may be able to spread those bacteria over long distances rather than just localized areas. So we do find resistant bacteria in all sorts of environments. But how do they get back to humans again, do you think? So do you think any or all of these are potential routes for antibiotic-resistant bacteria in the environment to spread back to humans again? So this is a sliding scale just to mix things up a little bit. So stick to the left-hand side if you disagree, and the right-hand side if you agree with these as possible routes for exposure. So yeah, we're all pretty confident that hospital stays are an issue. Bit more middle of the range when we're thinking about unwashed vegetables. So if there's a chance that antibiotic-resistant bacteria can end up in fertilizer and sprayed onto crops, then yes, there's a chance that we can pick up these bacteria again if we don't wash our vegetables and our salad leaves before consuming them. Yes, these bacteria can end up in all sorts of freshwater environments, including coastal waters. So many wastewater treatment plants are situated on coastlines because there's large bodies of water to dilute the treated waste into. But some of those bacteria might pass through the system and end up being released into the receiving waters, and those include coastal waters. So if we go swimming, bathing, surfing, there's always a risk, and that risk can vary, but there's a chance that we can pick up bacteria that way. and indeed through the air. So if you live in an urban area that's densely populated with lots of pollution, unfortunately, antibiotic-resistant bacteria or their genes can be carried on particulate pollutants, and we can inhale those. Hospitals are another place where we can pick them up, either through direct contact with other people or through instruments or surfaces, but also through the air. So sometimes hospital ventilation systems have been found to have antibacterial resistant bacteria in them. So unfortunately, these are all potential sources. And probably what you're realizing at this point is that it is a very complicated picture. That relationship between the environment and humans is very complex. And there are many, many routes through which we can be exposed to antibacterial resistant bacteria, but also we can spread them again to other humans. And there are a number of vectors. So we've mentioned birds and insects can carry these bacteria. And we can pick them up either directly through consuming water or being exposed to water, which may have become contaminated in the first place by us as humans or by animals, livestock, wildlife, birds, and so on. So it's a very, very complicated picture. So this creates a challenge for us as scientists to try to figure out what's going on in the environment. But Donald and I are really interested in trying to understand what's happening in freshwater environments. So how do you think we can go about looking at antibiotic resistance in freshwater environments? There are a number of options there. Don't worry if you're not sure, just say you're not sure. So can we test water for resistant bacteria? Can we detect them there? Can we look at DNA in environmental bacteria? Would that tell us anything? And can we measure the levels of antibiotics directly in water? Okay, so we're sort of an even spread really between all of these. It wasn't a trick question, but actually you're all right. We can do all of these things. And Donald and I are attempting to do that with our PhD student at the moment. We're trying to look directly for these bacteria in the environment, but we're also looking for the DNA that comes from these bacteria and looking at antibiotics as well. So we have a PhD student at the moment who's looking specifically at one organism called E. coli. You've probably heard of it before. It occurs in feces, so it's called a fecal indicator organism. So where there is E. coli, there is fecal pollution and probably other pathogens as well. And we're interested in one particular type of resistant E. coli that's called ESBL E. coli, and it produces enzymes called beta-lactam lactamases, extended spectrum beta-lactamases specifically, these enzymes can break down antibiotics, which means that these E. coli can become resistant to a lot of the beta-lactam antibiotics that we use to treat infections quite commonly. The original one being penicillin, that was the original beta-lactam, but there are new generations of antibiotics now developed from penicillin. And where we find high levels of this E. coli in the environment, then we know that there's high levels of resistance and perhaps a potential threat to public health. So our student is using what we call selective media, growth media that are specially formulated. They're chromogenic, which means that when we filter water and then place those filters onto the surface of the agar, the nutrients in there allow our E. coli to grow, and they will form a specific colour on the surface of the filter. And then we can count those colonies, and that tells us how many of those E. coli were there in the original volume of water that we filtered. And those media will have a particular antibiotic in it, so only the organisms that are resistant to that antibiotic can grow there. And we can start to track these organisms in all sorts of different water bodies across Scotland and get counts of these ESBL E. coli. And that starts to tell us something about which waters are most polluted. So which water source do you think might have the highest levels of ESBL E. coli that we've looked at so far? So we've got reservoirs, which contain water that goes on to be treated for us to drink, so potable water. And then we've got rivers and lochs, which are our natural still water bodies. So we've got a bit of an even spread again. Lochs not so much, but reservoirs and rivers about even. In truth, it depends. Depends where you test, but the chances are that rivers are most likely to have the highest levels of ESBL, E. coli. And that's because rivers tend to run through areas that are urban, where we have high human activity. Where we get high human activity, unfortunately, we get lots of waste. We get sewage waste, we get agricultural runoff, we get domestic waste, we get hospital waste, and all of these are carrying maybe antibiotics, but also resistant bacteria. And detecting these bacteria in water samples suggests that AMR is spreading through the environment and could impact human health. So we're particularly interested in rivers, but we're interested in all of these sources, and we want to make sure that none of these organisms are getting into our drinking water. And so far, our studies show good news, not getting into our drinking water. And that's all based on culture-based methods, so growing bacteria in the lab. But we've also got this really sophisticated tool that we call quantitative PCR, or PCR stands for polymerase chain reaction. Those of you doing higher biology will probably have heard of this. It's a way of copying specific bits of DNA, genes, specific genes, and copying them and multiplying the numbers up to a level that we can detect them. And we can use this approach to look at resistance genes in the environment. So we can take a sample of water, we can filter that and trap the microorganisms on the surface of a filter, and then extract all of the microbial DNA from that. And by doing so, then we can target in, and qPCR can be used to target specific resistance genes and make copies of that. And if we add in a fluorescent tag into that system, then we can monitor the increase in fluorescence throughout the cycles of PCR that are copying those genes up to levels that we can detect them. And the higher the amplification curve, then the higher the copy number of that resistance gene we found amongst the microbial DNA. And that then tells us that there was a higher number of those genes in the original environmental sample. So this is a great method because we don't have to rely on the growth of the microorganisms, which can sometimes be unpredictable and it takes longer. So this is a way to go directly to the DNA and detect the genes, the resistance genes directly. And we can look at lots of different environments. And we can tell a lot from that. So if we had a high level of an antimicrobial resistance gene in a water sample, what do you think it might indicate? So here's another question for you. So will it tell us there's sewage contamination, recent antibiotic use, or intensive agricultural use? Or is it all of these things, do you think? Great, lots of answers coming through. You're all right, of course, but it's potentially any or all of these. So if we've got high number of antibacterial resistance genes in a water sample, it tells us that those genes might be there free in the environment, ready to be taken up by a bacterium, or that those genes have come, we've managed to extract that DNA from living bacteria, but they're there, and they're there in numbers that we can detect. So what's going on in that water? It might be that there's lots of sewage coming from sewage overflow pipes or combined sewage overflows, which are used when we've got heavy rainfall to keep the wastewater treatment system operating normally and not above capacity. Unfortunately, sometimes that's quite common in Scotland, and untreated waste ends up being discharged into our rivers. You may have heard about this in the news recently, but it could also be that it's antibiotics coming from intense agricultural use. Okay, so there could be agricultural land nearby, or even wildlife. So it could be any or all of these things. And our job really is to think about the connection between the levels of antibacterial resistance, whether it's genes or living organisms, and land use. So we want to try to figure out the drivers for antimicrobial resistance, and to relate that to things like agricultural use, pollution, sewage, and so on. So that's what Donald and I do. But should we be worried? Well, Donald mentioned right at the start that this is a global health crisis. So yes, we should be worried. Can you remember the numbers that Donald spoke about earlier on? So he mentioned 10 million deaths predicted by 2050. So that's a lot. Yeah. So we're ranking what we think might be the highest cause of deaths by AMR by 2050. And you all think the AMR might be the highest one. Okay. So actually, it's probably going to be heart disease. That's still the number one. Sorry, it's not ranking it for me, but heart disease and cancer will probably still be out there in the lead. But the chances are that between now and 2050, the deaths from heart disease and cancer may double at most. But the deaths from antibacterial or antimicrobial resistance may increase fivefold between now and 2050. So that's why it's so concerning. It's the rate of increase that we're worried about. And I just want to show you this histogram. It looks very complicated, but it's not really. This was a study that was published in the esteemed journal, The Lancet, just last year, 2024. And it was work undertaken by a global network of scientists who looked at data relating to deaths caused because of antimicrobial resistance. So in other words, people being treated for infection and it not working, and them dying as a result of this, either directly, so directly attributed to AMR or associated with AMR. And they looked at this and they divided it by age, okay, by age group. And they compared the death rates in 1990, which are the green plots, versus 30 years later in 2021, which are the blue plots. And what they found is that the trends vary significantly with age group. So for young children up to about the age of 15 to 20, the rates of death due to AMR have decreased in the last 30 years, which is good news. But unfortunately, for young adults from about the age of 25 upwards, the deaths from AMR, either directly or indirectly, have increased since 1990. And it's this data that's being used to predict the trend working towards 2050. So the World Health Organization looks at trends like this to predict how many people may die because of AMR by the year 2050. And what we can see is that particularly in older ages, the chances of dying as a result of AMR increase by up to 80% for those who are age 70 and above. So it's age-related, but even for young adults, the risks are increasing as we go forward. So what are the impacts from all of this? So which of the following do you think is unlikely to be a consequence of rising antibiotic resistance? You pick your answer. So increased hospital stays, recovery time, increased treatment options, and increased economic costs. So which do you think is the most unlikely to be a consequence of increased resistance? So increased treatment options seems to be the preferred choice, and you're absolutely right. Yes. So we will have increased hospital stays, increased hospital numbers, increased recovery times, maybe less full recoveries. And this all has an economic cost for society and for healthcare systems as well. But the one thing it won't give us is increased treatment options, not unless we take action and we find new drugs. So maybe as future scientists, that's what you might do. You might go on to explore different antibiotics or alternative treatments for antibiotic infections, or you might go on and do research. But the one thing we won't have if we do nothing are options for treating multi-drug resistant bacteria. So at the moment, we have last resort antibiotics like vancomycin that are used normally only when an organism that infects us won't respond to any other treatments. But the problem with these last resort antibiotics is that they often have serious side effects and they're very expensive antibiotics, and we want to limit their use because the more we use an antibiotic, the greater the chance of resistance developing. So they really are our last line of defense. So what we need are new drugs, alternative drugs to help us treat this. So in the meantime, what can we all do as normal human beings, not as scientists? What can we do? So we can think very carefully about the antibiotics we use. So if you were prescribed antibiotics, what would you do? So this is a sliding scale one. So you can either disagree, stick to the left-hand side, or strongly agree with each of these statements. So would you stop taking an antibiotic once you feel better? Do you disagree or agree with that statement? Okay, so you can answer all of these. Would you share them with a family member who has similar symptoms? Would you complete the full course, even if you feel better? And would you discard unused antibiotics into the recycling? Okay, things are still shifting a little bit, quite different answers for each one. So stop taking them when you feel better. You're sort of middle of the road there. So this is a really important one. Never stop taking antibiotics when you feel better. So always follow your doctor's instructions to complete the course, even if you do feel better. Take every last antibiotic. Yeah, and never share your antibiotics with family members. Even if you know they're infected with the same organism, they must get their full prescription. So you're being prescribed a dose that makes sure that we clear everything from your body and doesn't leave any resistant organisms behind. And please don't flush your unused antibiotics down the toilet, because you're perpetuating the problem of antibacterial resistance in the environment. Make sure that you return unused medicine to the pharmacy or to your GP surgery. Okay, so that's what we can all do. Thank you so much. Well done for answering all of these questions and taking part. Hopefully, you've learned a bit about antibiotics today, and you can share what you've learned with your family and friends and bust some of those myths for them and make sure that they take antibiotics correctly as well. And thank you very much for taking part today. There are lots of other resources, by the way, that you can search. So the UK government, Antibiotic Research UK and the World Health Organization all have lots more information if you are interested. And thank you for your attention.
Thank you both very much. We have had a few questions come in, so we'll get started on those just with the last 5 minutes. Starting with your top tip, for those looking to get into medical research, what would be your top tip to pupils? Putting you on the spot.
Shall I go first? So, because I didn't go into medical research, I can't answer that really specifically. But of course, I think you know, it's research is research in a lot of ways. So top tips, I think the first thing for me is really figuring out what you're interested in. And that's really difficult when you're a school pupil. You're studying really broad subjects. And so I think, you know, unless you're really confident about a specific area within medical research that you're interested in, you probably want to start with a nice broad undergraduate degree. And pay attention to what you're really switched on to, not what your friends like or who has the nicest lecturer and is kindest to you and gives you the highest marks, but what you really think you're passionate about, because you're going to have to stick with this a long time through thick and thin. And you have to also be able to express your passion for that subject to other people. You're going to work with students and colleagues. And so I think having a real passion for something is key, and that will give you the resilience to stick at something no matter how hard it gets. That would be my top tip.
And for me, the route I took was through working in the hospital environment. So there are a number of different jobs that you can aim for within the hospital environment. Within microbiology, there's a large hospital microbiology laboratories in every city, several of them. So you can train to be a biomedical scientist or a clinical scientist, which is the route I took, or a medical microbiologist with a medical degree. So these are the three options for you to aim for within the hospital environment, and that then exposes you to an environment which will lead to many different areas of medical research.
Perfect. And is there any subjects aside from science and STEM that you think would be useful for pupils?
Lots of subjects. Having a broad skill set is really important. One of the biggest things we have to do as scientists is communicate, and to communicate with different people. So it can be members of the public, or it can be people who are stakeholders who might influence change. So we might want to talk to ministers, or we might want to talk to doctors. And you have to have a different style of communication with all these different people. You have different language, different vocabulary that you use. So your English skills, so your written and your oral communication skills are really important. I grew up chronically shy. It's something that I think a lot of school pupils feel, they're terrified about maybe giving presentations. The more you do it, the better it gets. So I think your English skills are incredibly important, but I think there's lots of scope for creativity in science. So for me, I loved art at school, and I think, what it's helped me do is create visual representations of what are quite complex scientific ideas. So I think a broad skill set and play to what you're good at. So if there's something else that doesn't seem immediately related to science that you're good at, think about how you can utilize those skills and incorporate that into your style of science, I guess.
And I have nothing much more to add to that than to say, there are many routes and there are many directions you can go in, and there's always a possibility of changing at different stages. You're not trapped. Even after doing your first degree, or even in the middle of a degree, you can change to different disciplines. So you're never trapped, always open to change.
That's brilliant. And ending on a positive, what's been the highlight for you both in your careers so far?
So I'll come first on that one, Fiona, if that's okay, because for me, I was just thinking of that as I was giving my talk. I mean, research is just exciting. I mean, I spend much more time on it than I should, evenings and weekends, but partly because it's busy, of course, but partly because I just really enjoy it. So I'm thinking about the research project. We've got PhD students and which direction to go in. So just the excitement of it. And the other thing about being in research is that when you do some work, then you need to present it to different people in your field, and you get to go to different conferences. Obviously not very often, but I've traveled around the world to a number of different places that I would have never got to if I wasn't doing research.
Okay.
Yeah, similar to Donald, I think discovery and being able to share something is really nice. And what conferences do, though, is put it all in perspective, and you realise how small, what a tiny piece of the puzzle your work is. And it's important to keep that perspective. But that sharing of knowledge with other scientists is always really fun. I think for me, though, I think it's the day-to-day seeing students get excited about subjects and relate that to the real world, and seeing them just switch on and say, right, I get how this is relevant to the world. I get how I can actually, however small my contribution is, I can make a genuine difference. And to see them go off, whether they've done a PhD or a first degree, it doesn't matter to me, but seeing them go off and start a career in something related to this, for me, is definitely always the highlights. Yeah.
That's lovely. Thank you both so much for joining us today, and great answers there as well. And thank you everyone for joining us. And we've got our last and final Medical Research Scotland Academy next week. Do make sure you're following the Medical Research Scotland YouTube channel to keep up to date with the Academy. And thank you again, and we'll see you next week.